Hi lads, I wanted to do genetics with you today, so I'm just going to spend, it's probably going to be about 30 minutes long really, on genetics. We're going to look at all of the key kind of things we looked at so far in terms of sex linkage, in terms of dihybrid crosses, and the question actually that you'll have for homework really for me is incomplete dominance as well. So we just want to go through that as quickly as possible, okay? So this, as you know, genetics comes up almost every single year. It's really something you need to know inside out. This question here, sex linkage, I know sex linkage has caused a few problems for us, but we're just going to make sure we understand it now, okay? So the human characteristic of red-green color blindness is governed by sex linkage, okay? So we're talking about color blindness. The allele for normal vision is dominant. So of course normal is dominant, so we get a big N for that. The allele for red-green color blindness, so we have a recessive allele there. Okay, so the first thing it says to do is explain the underlined terms. Actually, the underlined terms in this question were dominant. Okay, so dominant, remember, is where the gene is expressed in the heterozygous and homozygous condition. Okay, and the other underlying term from the question was sex linkage. Okay, so sex linkage is where the gene is found on the X chromosome. Okay, so that is the first question there. Okay, apologies for the writing here. We're out of printing cartridges down here. Using a diagram determine the possible genotypes and phenotypes of the offspring from the following cross. So we have red-green color blind male. Okay, so male anyway we know is going to be XY. And if we have red color blindness, then we know we're going to have a recessive allele there, which is going to be small n. We have a female, it's going to be XX. And if she is heterozygous, then she's probably going to have a capital N for normal and a small n for recessive. Okay. And the final question asks us to give another example of a sex-linked characteristic in humans. So another example other than color blindness. Okay, so I'm going to do that question now below here. So the first thing you do when you're doing any type of genotype cross is you write out the parent genotype. Okay, so the parent genotype in this question, well, we have a male who's always going to be XY. And from the question, we've already worked out what his situation is, and is as regards vision. Okay, so we're told that using a diagram, possible genotypes and phenotypes of offspring from the following: red-green color blind male. Okay, so therefore we can put, if we know he's red-green color blind, we can put a small n on this. Don't forget here we have a line coming down, but actually because sex-linked genes are carried on the X chromosome only, the Y chromosome doesn't have any allele for vision. Okay, so that is the male. We can call that the male. We next have the female. Okay, so they're reproducing, and the female here is going to always be XX, because those are the chromosomes for a female. And we know that this female has heterozygous alleles. So because she's a female and she has an X chromosome, each of her chromosomes is going to carry one of these alleles for vision. Okay, she's heterozygous, which means she has different alleles. So we have capital N and we have small n. And this effectively is our genotype cross. We're going to multiply, not multiply, but rather we're going to cross these two together and see what happens. Okay, so if you think about there, first thing we did anyway, we got the genotype of parents. The next thing we need to work out there, and which is essential, is the gametes. Okay, so the gametes. So this is really important to remember. A gamete could be, for example, it could be talking about a sperm cell, okay, or an egg cell. Now remember, these are haploid, okay? They have half the chromosome number, and that means that they only get one of these alleles, okay? So when we talk about possible gametes here, for example, the male, in one of the sperm, he could pass on X with small n. In another sperm, he could pass on Y with nothing, okay? And they are the possible gametes for the male, okay? The female could pass on X, big N, or X, small n, okay? And remember, in an offspring, in an actual human being, you've got to have a pair of alleles. That is a fact, okay? A pair of alleles for every characteristic. Whereas if we're talking about sex cells are only ever going to have one of those alleles, okay? They'll never have two, because remember, the sperm cell and the egg cell have to fuse, and then they bring the two together, okay? 1 and 1. So that is key there, because I'm starting to see an awful lot of confusion as regards the gamete and the actual offspring. This is just the gamete, okay? There is no offspring yet. But when we do a genotype cross on this, we will see that we have got offspring with 2 alleles, okay? So now we're looking for the offspring, and we're looking for the offspring's genotype. So if we look at the genotype here, big X, small n. Okay, big X, small n could go with big X, big N from the female. Okay, so I'm going to write that. Well, do you know what? We'll just keep it, we'll fix it in a second. We'll just write it big X, big N. Okay, next one, big X, small n again, could go with big X, small n. So do you see that? Big X, small n went with big X, big N from the female. Big X, small n from the male went with big X, small n from the female. So we've now covered both of those. As well, Y chromosome could go with the first one, which is X, big N, and Y chromosome could go with X, small n. Now, the way I've written out this, the order is completely wrong, but I just wrote it out that way so you can just see it maybe a little bit easier. Okay? The order, you should always put the dominant allele first. So X big N should come first, and then X small n. We have X small n, X small n. So it's XY, so it should be XY, so therefore we have it as such. We have again XY and should be small n. Okay. Now, the next thing you need to do is you need to work out your phenotype. Okay, so we've got the genotype. We have 4 different possibilities, okay, so 4 possibilities. And that's for the genotype. So the next thing there is the, so we've worked out the genotype, we have the genotype, so therefore we want to work out the phenotype. So remember, the genotype is just the genetic makeup. So this is the genetic makeup here of the offspring. The phenotype is going to be what it looks like. This is going to be a female because we have 2 X chromosomes. This is going to be a female because we have 2 X chromosomes. This is going to be male because we have XY, and this is going to be male. If I look here, I have big N, small n, so that is going to be a carrier. That means that she has got the allele for color blindness, but she has a big N, which means she has normal vision. So normal vision is dominant to small n, so therefore she's a carrier. She carries this gene, but she does not suffer. Okay, female, if you look here, female colorblind, because we have 2 recessive alleles. Male normal and male colorblind. Okay, now a question that often comes up on this as well is why are males more susceptible to color blindness than females? And that's because males will suffer, females, if we talk about females first maybe, females in the heterozygous condition are only carriers, whereas if males get one of these alleles, they will be a sufferer straight away. So that's why males are more susceptible to color blindness, and it's also why there's an awful lot more males who are colorblind than females. There's a massive percentage of males compared to females. Okay, the final part, if we go back to it, it asked, give another example of a sex-linked characteristic in humans. So for that one, you could just maybe pick out hemophilia. Hemophilia is a nice one to remember there. Okay, so that is sex linkage. Okay, the next thing that I want to do is go over your mock paper question.